Good morning, guys. Thanks for braving the storms or storms to be. Everybody hopefully has an umbrella. Oh. Yeah, I saw you coming a little bit. Well, then you're even more of a, hardcore drug discovery lover. So today we're going to talk about something that most of us are not that exposed to, which is preclinical toxicology. And I think we have a really wonderful speaker today, Lucia Miller. So she's actually from Southern Research. So as many of you know, we have a collaboration with Southern Research, but I mostly work with the Preclinical Drug Discovery Division. There's a whole other division we work with sometimes, but not as much, which is the Drug Development Division. Well, and then there's engineering, and they do stuff in space and things like that. I don't know much about that. But the Drug Development Division that Lucia is part of, they do a lot of contract work for actually outside entities that have their toxicology studies done at Southern Research. So they're very experienced there. So Lucia is a certified toxicologist, so I'm really excited to have her here and kind of talk about what things you need to think about when you design a toxicology assay or experiment. So here you go. Welcome.
Well, thanks everybody for coming to my talk. I hope you're revved up and excited to learn about preclinical toxicology. So I'm just going to give an overview, what is toxicology, why do we do toxicology testing, and just give some kind of historical examples to kind of give you a feel for how important it really is. So there's just a brief outline of the presentation. So first things first, toxicology is just the study of the adverse effects of anything that's chemical, physical, any biological agents, biohazards, biowarfare, even vast amounts of, even say water. If you have a glass, you're fine. If you drink 20 gallons, you're probably not going to be fine. And it's just the effect that all of these things have on either people or animals or environment, and really what you're studying in terms of, if you're going to be looking at, say, people, you can look, you may be in forensic toxicology. You can be in environmental toxicology if you're looking at, risk assessments for, levels of boron in the water or lead or something like runoff in the dirt. So it really just depends, and it's all kind of grouped together like this very broad field, but there are these subdivisions depending on what area you're studying. So really everything that we eat, breathe, come in contact with, anything, even the food on your plate, the chair that you're sitting on, really any of those components can have a toxicological effect. And I don't mean you're going to sit on your chair and you're going to just speaking more broadly. And so, as I mentioned before, really everything, including water, again, it's all about the dose, right? So the whole, if you take nothing else with you today, it's that the dose makes the poison. Just remember that. So, you can have, if you think back to, I don't even know what that period of time was called, but when, there were a lot of poisonings, and they would, give these small amounts of poison over time, and then they would just to build their own sensitization, and then they would, kill a king by giving it the poison, but then he would also have the poison in his drink, but he's been sensitized to it.
So, like in the Princess Bride.
Yeah, like in the Princess Bride, exactly. Yeah, so, that's just an example, kind of, in that case, that same dose of that poison was toxic for that king, or that unfortunate individual, but not for, the one who had desensitized himself. So as I mentioned before, it's really just the interaction of how these chemicals or biological agents interact with your system or the environment, and what outcomes they have. And so it's a very integrated, it's a very, very broad field. So why do we even do toxicology testing? So I'm at Southern Research, as Michael mentioned, we're a contract lab. And so any company that wants to develop a drug, say, you have your aspirin or your Tylenol or any of those medications, they don't just go from a lab to the shelf. They got to make sure that it's going to be safe for you. They need to know how much they can give, what the dosing regimen is going to be, and if there is any toxicity, you need to identify it and characterize it so they know, okay, well, this compound, is going to cause liver failure. Well, they're not going to give it to you. They're not going to let it go on the market, right? So anyway, really, that's the whole point of preclinical toxicology testing. And so if we're looking at this little scheme here, so you start, as the drug discovery part where you have like thousands of compounds, and then as you move forward here to the preclinical phase, which is where you have, your animal models. This is kind of the space that I'm working in. But really, I mean, just kind of to take a bird's eye view, the process is pretty long and it takes a lot of money. It's very expensive. And so really sponsors are not going to want to get to, phase 3 clinical trial only to realize like, hey, this causes kidney failure. And then they lose all that investment and then they have to go back to the drawing board. So really the goal of the preclinical testing is to try and evaluate what potential toxicities of that novel drug compound are. And you can use, different animal models, which I'll talk a little bit more about in terms of, how you select the animal models, because some are better than others. So right now, we do have some computer simulations that you can use to identify certain structural alerts. If you have a benzene ring in your compound, you're probably not going to get to market. So it's pretty carcinogenic. So we'll use, animal models for now until we, maybe later in next lifetime, identify that all in silico. But for now, we use animal models to try and replicate, not replicate, but represent the human situation, because you can't just give, drugs to humans, which there was a time when you could, when they would just test them on humans, but we don't do that anymore. I say we, I had no part of that. But so with the animals that we use, they're just, for anybody that, there are very strict guidelines for the use of animals. You can't just order a bunch and just give them all this drug, and, like you have to explain to, we have an institutional animal care and use committee. So anytime we want to use any kind of animal for anything, we have to write an animal care and use protocol. It's going to say we need this number of animals for this purpose. This is what's going to happen to them. Then the IACUC is going to review that and say, you're saying that you're going to do this frequently, but the guide says you can do it only this frequently. So you have to revise the study design, really, because you're just trying to think about animal welfare, right? I mean, you want to get your drug to market. That's great, but they're also animals, and you need to take care of their welfare. So. Really, and again, depending on what your endpoint of interest is, that will inform what kind of animal you use. So for example, if you have a topical cream you want to test, you're probably going to use the pig, because their skin is the most similar out of all the other species than any of the other species. So it would be more closely representative of the human situation. And the point I like to stress is that it is very, very important, especially in the case I'm going to present to you in just a second. Now, has anybody heard of thalidomide, or the thalidomide tragedy, thalidomide disaster? And so back in the 60s, they, this German company, Preclinical, I think, marketed thalidomide as an anti-nausea drug, worked really well, and women just, it was very effective. And when they did the testing, they saw nothing. They said, okay, great, this is very effective. It causes no malformations. We're good. And so what ended up happening is that babies started being born like this. And this really is touted as probably one of the largest, drug disasters of our time. Essentially, what happened is that when they did the preclinical testing, they tested it in rodents. So they gave upwards of 1,000 milligrams per kilogram. Nothing happened. They were fine. But then what they ended up doing is they tested it in rabbits. And the rabbits, they saw the same outcome. And so that's why the FDA now requires that when you do your preclinical safety testing, you have to do it in a rodent species and a non-rodent species, just to make sure that you capture the effects, because there can be some pretty serious, pretty large species differences in some of the outcomes that you see. So just something to think about. And that is called phocomelia for anybody interested in the terminology of that birth defect. So again, there are some key assumptions that you're making with your animal model. And really, with any model that you use, if you're going to use a chicken embryo to try to represent a human child or embryo, or if you're going to try to use a rabbit to represent the human situation, you need to just understand the strengths and the weaknesses of the model. Period. I mean, the better you can characterize it, the better you can interpret the data that you get from that model. Because if you, for example, if you were going to use a rabbit strain that in a developmental reproductive tox study, and this particular strain of rabbit aborts 40% of the time. You know, you need to know that because one, you're gonna have to account for the number of animals that you're gonna use. And two, if you see that level of abortion, let's say, unfortunately, just in the high dose group, you'll know that, you know, historically this strain has a very high rate of abortion. So you can kind of help characterize it, you know, just to understand. I'm not saying that you wouldn't have to look into that some more, but just ways of kind of understanding what your model is actually telling you. So one of the things that was probably the biggest learning curve for me going from academia into drug development or contract lab was learning good laboratory practices, or GLPs. So these essentially are, it's a code of federal regulation, and it outlines, okay, how you're supposed to conduct your laboratory study, not in terms of study design or anything like that, but you know, how you document things, the fact that every study has to have one study director assigned to it, and there's a single point of control, and you have to be able to characterize. So let's say, you know, I'm the study director for a study. Sponsor wants to test their new drug. When they send us their, they call it a test article, they need to, or we need to somehow, we have to have data that will characterize what it is. So identify it, say what the purity is, its composition, concentration, so that you know that what you're giving to that test system or the animals is actually what you're giving them. And so that's just things like that are required in order to be in compliance with GLP. So it's, you know, FDA for the US, and then there's OECD and EPA as well. Just they're similar, but not identical, but they're similar. So any definitive studies that are done, those are the ones that say the sponsor will use to submit to the FDA to say, okay, this is the outcome of the study, the drug is safe, can I go into the clinic? I mean, it'll be a multitude, it's not just gonna be one study, it's a panel of studies that you do, which isn't always the same for every compound. It depends on what the indication is, it depends on what type of molecule you have, because a small molecule, just, you know, a basic chemical is going to be different than, say, a biologic or an antibody or a virus or a gene therapy or something like that. They have different requirements. So why GLPs, if you were just burning to know? Back in the 70s, there was a lab called Industrial BioTest, and you can Google this, and it's sure the first thing you'll do when you leave here. Essentially, they were falsifying data. They were testing compounds, submitting them for approval. They would basically not report if animals died. And if you look in the data when they did the audit, there were animals that died and then later showed up in the data again. And so, I mean, you know, they ended up calling that lab room the swamp because the humidity was so high that it was just like waters on the floor. And then there were animals that were just like in their cage. It was a bad situation. So anyway, the people that did that went to jail. And then the FDA said, okay, no, we need to regulate this. So now any pharmaceutical compound that you have on your shelf has most likely gone through pretty rigorous safety testing, exclusive of, say, things that like GNC or nutritional supplements that don't go through testing, which is still beyond me. So in vitro toxicology is typically where you're going to start, and it gives you a lot of mechanistic information about your drug. You know, if you can test in hepatocytes to, you know, to represent liver cells, or you know, just various types of cell lines to see, okay, is this a mutagen? Is it you know, is it genotoxic? And in a non-animal model, because you can get a lot of information from these assays without using animals, and they're way less expensive and they're faster. So it'll just help also kind of weed out some candidates if you have, as I said, you know, 10 to 15,000. That's just a general number. It's not all the time, but just help to weed it down so you're not dealing with this whole handful of molecules that each of them is going to go. Yes.
So how likely is in vitro toxicology assay give you false negative results?
Well, that largely depends on the assay itself, because I mean it depends on the molecule and it depends on the assay sensitivity. So I mean, if you're looking at something like Ames, or if you're going to look at something like, you know, like rodent whole embryo culture, it is really assay specific. But I mean, that's the whole reason. And I think, you know, sensitivity and specificity of the general assays that are used preclinically in a battery, mind you, are around maybe 80 to 80 or so %, but they're done, as I said, in a battery, and it's just to give you information. It's not going to be definitive. You know, you see a positive result in the Ames assay. Oh, this is carcinogenic. Throw it out. Like, you know, you have to do some other test to characterize it, but it's really just a battery that you use to kind of help inform. And then, you know, as a follow-on to those assays, you know, let's say you have the 2-year chronic carcinogenicity study in animals, which may show no cancer, apart from baseline. I mean, you know, you keep them around that long, they're going to develop tumors. But, you know, so you may have results from one assay to the next that don't really match, but, you know, you've got to kind of take the whole, the weight of evidence approach. So it's really on a case-by-case basis. So as I said, you know, you can screen chemicals for different types of toxicities. I keep referring back to Ames just because it's one of the more widely known tests to test for mutagenicity to see if it can take DNA. You can look at, depending on the types of cells that you're using, if you want to look at liver cells, skin cells, you know, brain cells, you can look at those as well. So it's pretty versatile, and you can have high throughput, so it's a nice speed to get the data. And also less expensive because none of it is free. Mm-hmm. And they're faster, and they don't bite, mm-hmm. because some animals do. And but again, you know, the big kind of elephant in the room with these tests is that it's, you know, less predicted than a whole animal, because you don't have the whole physiology, you don't have the whole system. So I mean, if you're going to try to determine if something is, you know, can cause birth defects or is teratogenic, you probably don't just want to use, you know, one cell type, because you have, you know, the maternal system, the embryo, it's 2 systems, like, and they're interacting. So you want to just try to use something more representative. And so this is just a list of, you know, different things that you can screen for in different in vitro tests. You know, if you're looking just for cellular toxicity, the way that protein binds, kind of just understand more mechanistically how is this drug working. And this is the in vitro part, again, is done before or sometimes in concurrence with the animal work. So this is kind of before it comes to us. So in terms of cytotoxicity, you can look at things that affect cell structure, you know, like membrane integrity. Does it just completely dissolve the membrane? That's probably not a good thing. You know, function, if it's gonna, you know, impact its ability, the cell's ability to turn over oxygen or you know, metabolize or synthesize DNA, which probably isn't good either. But you can use a wide variety of cell types, and again, it's quick and it's informative, but again, the model that you select is very important. So it's always good to just keep that in the back of your mind. So in Europe now, you cannot test cosmetic products in animals. So what they would do is there's a test called a Dray's irritation test. We still do it here, not as often, but that's more so like skin irritation, but they would actually put the compound in the eye of the rabbit and then evaluate irritation, which, I mean, you can imagine is probably pretty uncomfortable for a compound going right into your mucous membrane. So Europe said, nope, you're gonna do this in vitro. And so, you know, they have something like epiderm. If they're looking at toxicity to the skin, they're gonna use skin cells instead. So that way, you know, it just saves that poor little rabbit some pain. But a lot of these, thankfully, are available in kits. And so they have nice instructions, and they tell you what to do, and you can have your standard curves along with it if it's on a plate reader. So it's, you know, you get pretty decent results with those. In terms of interspecies comparisons, you know, there's a lot of words here, but really, I just, again, stress the point of different species and even different strains of the same species can behave differently. So I do a lot of DART work, or developmental and reproductive toxicology work, and one of the most important things you need to have is a historical control database, because if I'm going to use the Harlan-Sprague-Dawley rat, I'm going to need to know its rate of malformation, what its incidence of abortion is, or resorption it's called, or what its incidence of, you know, non-pregnancy rate or pregnancy rate, you know. Just things like that, because if I'm going to then switch over to a Wistar-Hahn rat, they're not going to be the same. And so you just need to be able to characterize those things. So the importance of all of the preclinical testing that we do is really just to establish a safe first in human dose, period. all the testing that's done, your in vitro tox tests are going to inform the dose that you start with in animals. And then all of your animal testing is going to inform the start dose that you're going to with humans. So it's kind of just like a graded approach, right? So just to try to mitigate risk, so you don't go into the clinic with a dose that is just, really just toxic. This typically doesn't, there's a lot of good characterization that happens beforehand, so it's rare that happens, thankfully. But that's really that's the take home there, is establishing that safe starting dose for clinical studies. And you want to know if there is target organ toxicity. Is it going to target the lung? Does it target the heart? Does it target, so you'll do a full panel of tests and you'll do histopathology on the different organs to try to see what the toxicity is and characterize and know at what dose does that happen. Because if you're going to start in a study in animals, if you go up to 100 milligrams per kilogram, you have to scale that back tenfold when you start in the clinic. And you scale back from a dose that caused no toxicity in the animal. So these are just some of the types of preclinical tests that we do, over at Southern and other CROs and pharma companies that the FDA would require data from in order to move your drug candidate along. So one of the first things is a single dose test. Is just give it once. What happens? You observe the animal, and you take all these endpoints and observe, its behavior, more like clinical signs. If it starts, seizing or something, that's not good. So once you have your single dose, then you can move on to repeat dose. And typically with your single dose, you're also going to have some pharmacokinetics. If you're going to draw blood and evaluate, like, what's the pharmacokinetic profile of the compound, so you know how often you can dose. Because if you have a drug that has a half-life of a week, you're not going to dose every day because it's just going to keep building and building and building. So you want to know that information. So before you do your repeat dose, which is typically done in compliance with GLP, and GLP typically takes longer and costs more because you have a quality assurance unit that audits pretty much everything. And then, as I said before, the TK and PK studies are just to evaluate where's the drug going, how much of it, is going, and how quickly does it go away, basically. And safety pharmacology is just a panel of tests to evaluate the impact on the respiratory system, your nervous system, and your cardiovascular system. And those are pretty important systems to keep you alive. So those have to be done just to demonstrate that your compound's not going to cause a torsadile decline or just cause you an arrhythmia or something like that. So you just want to kind of weed those right out because those are no-goes. And then genotox testing, as I mentioned earlier with the Ames assay, just because you get a positive read that in the Ames assay doesn't necessarily mean your compound's going to stop there. You have other tests in animals, like a chromosomal aberration test where you dose an animal and then you take out from their bone marrow or from their blood cells and look to see if their chromosomes, what happened there, if there are any changes there. So it's a little more reflective of an in vivo situation. So one of the things that I learned over time is that no two drug development programs are the same. They're all different. As I said before, depending on the type of molecule it is, depending on what it's indicated for. So for example, if you have a drug that is going to treat disease that is so life-threatening, there's going to be not less to do, but that's taken into consideration. If it's the only drug to treat this disease, or if it's a drug that is going to be given for a disease that is only in people aged 60 or above, they're not going to worry about developmental and reproductive toxicology. So they're not going to need that testing, right? It's just kind of, well, what do you actually need it for? Who are the patient population that this drug is going into? And then you kind of focus on that. If you're going to have a drug for prostate cancer, you're not going to give it to a woman. So it's happened. So as I mentioned before, those animal studies are typically done in 2 species. What I mentioned before about the rodent and the non-rodent for DART is usually the rat and the rabbit. And then for general tox, it's typically the rat or mouse, depending again on the compound, and the dog, or the monkey, if there's like a justification or need for it. That's typically in cases of, if you're going to have an antibody or something like that, and then the NHP tends to react closer to, you know, the human. Whenever the immune system is involved, typically that's when you're going to go with NHP. So as I said before, you typically will start with your single dose. These studies are just, again, identify where can we start that's, you know, that's okay. And this is typically not GLP. So when I say not GLP, it doesn't mean just, you know, throw the rules out and just dose the animal and then leave the room. I mean, you still have to do those documentation practices. You just don't have the QA unit auditing you. So we still do everything according to standard operating procedures that are basically our interpretation of those GLPs. So the way that we document things, the way that, you know, we record data, the way that, so things like that are all guided by SOP. We just don't have another person auditing it. These are pretty short studies and they don't include really a lot. I mean, you just kind of want to understand if you're seeing any kind of toxicity at this point. And if you're not, then, you know, you can move on with your repeat dose. As I mentioned, with the small molecules, you have your 2 species. And with a biologic is, you know, because you can have a potential immune response, and also because it doesn't behave the same as a small molecule, you don't always need 2 species, because you may have that biologic that will react in one species but be completely inactive in another one. And so there are these guidances, the International Conference of Harmonization, or ICH, that kind of give some guidance. They don't tell you exactly what to do because every drug's different, but it just gives you some guidance in cases of when you're really not sure. Ideally, you would want your drug administration, like how frequently, what route, like if you're going to give it oral gavage, you're going to give it IV. Ideally, you want it to match what you plan to give into the clinic. Doesn't always happen, but that's, you know, ideal. And so the kinds of things that you look for in a repeat dose study are a lot more extensive, just because you have more drug in that animal. So you want to understand, okay, well, what target organs does it affect? We're going to look at blood parameters, clinical pathology. Does it impact, you know, are they losing weight? Are they stop eating? Are they not throwing up, or don't throw up? But you know, and so when you have, especially with, you know, say like NHPs, because they are they're a regulated animal also. So I mean, you want to try to get as much data from them as possible. And so, you know, you can even do heart exams typically on the monkeys and the dogs as well. And what you're looking for when you select these doses, that you're typically going to have four dose levels. One is going to be the vehicle, so 0. You're going to have a low dose, mid dose, and high dose. And what you want to see at the low dose is pretty much no toxicity. Mid dose, you want to see some toxicity. And at the high dose, you want to see toxicity, almost like an extension of what you saw at the mid. So you just kind of see this dose response. And the reason why FDA wants that top dose to cause toxicity is because then they know that you span the entire dose response range of that compound. So you're not going to get any surprises when you know, if you saw nothing at the top dose, they're going to say, well, you didn't push the dose high enough. You have to increase the dose so we can see what the effects are. And I mean, you're not going to give that high dose to patients. It's just to understand what the range is. So as I mentioned before, the safety pharmacology, or just, you know, to evaluate those safety of those important systems. Chronic tox and carcinogenicity. There is a lot of push to move away from this type of study design because it's long and just very, very long. And it takes a long time to I mean, it's a 2-year study. The rats are based, you know, you dose them according to your dosing regimen, and then they stay there for 2 years. And I mean, they'll naturally get tumors. I mean, they'll also get really fat, and I mean, they just, you know, then the necropsy at the end of it just takes forever. But, you know, it gives pretty good information about, you know, rate of carcinogenicity. But they're not done as frequently as they used to. I think they're trying to move away from that study design because it's so laborious and involves so many animals. And then reproductive toxicity and trigenicity, which is near and dear to my heart, evaluates toxicity in pregnant animals. And so you can look at it at the various stages of the life cycle of the animal. So, you know, you can have fertility tests where you give the drug during, you know, spermatogenesis, or, you know, during, you know, basically during sexual maturation, and then you mate them and see, okay, do they get pregnant, or is it an effect on the sperm? Or you can look, you know, for trigenicity, like birth defects, like for that thalidomide case, that would have been an embryo fetal development study where you get we order time pregnant animals, and then we dose them during creative organogenesis, which is essentially when all the organs are forming structure. And then if you see birth defects, it's not good. But anyway, but you can evaluate that, and then you can also look at things like behavior postnatally, and there's a lot of neat things you can look at. So biologics really they're quite different than small molecules. They have different requirements. They behave differently. For example, you can have a drug as an antibody, and your animal model will develop anti-drug antibodies, which can be toxic in and of itself. So that's just different things to consider when you have that type of molecule. Because of different species differences in the way they react to different biologics, often you would use either ferrets or hamsters or cotton rats, which are just mean little things. But, just calling back to these key assumptions up here, really, you're just trying to get a predictive model, but you need to know the strengths and weaknesses of that animal model. As I said, different animal models can react very differently than humans. As I said, with thalidomide, that's just a perfect example. And now the FDA is moving more towards having sponsors, show them some preliminary data of, well, why are you selecting this animal model? Tell me, justify why you're starting with this one. So they become a little more stringent about that. And these are just, a variety among many why different animal models may be completely inappropriate. They may not have the receptor that your drug is trying to target. They may have the presence of a target that doesn't matter. And I'll give a couple examples after this. Or they may, handle the drug differently. They metabolize it differently, either really quickly or really slowly. And then that, if they bioaccumulate, that's not going to be relevant to humans if humans get rid of it much faster. Susceptibility to infection. So when you're involved in the immune system, that's kind of where it gets a little bit more complicated because the immune system of different species can react quite differently. As I said, cotton rats and hamsters for adenovirus vectors, and they're just really, really mean. So for example, I mentioned earlier about, the prostate cancer, that you wouldn't want to test a male-specific drug in females. There's really no point. I'm just going to skip over that antibody example. But this example was one of the first ones, and one of the ones that was always kind of driven into, every intro tox course, is the example of unleaded gas. If you test it in rats, they're going to have a pretty serious nephropathy. They're going to have a lot of kidney toxicity that you don't see in humans. And so when this was investigated, rats have an enzyme that will generate what's called alpha-2 euglobulin. And they're just these little goblets that basically, get stored up in the kidney, and then they will burst. So that's no good. Humans don't. We don't generate this, and so that toxicity doesn't happen, so it's not relevant for humans. So understanding those kinds of species differences will really help to put that into context. Tamoxifen, it shows genotoxicity, meaning that it can mutate DNA in the livers of both rats and mice, but it only causes cancer in the rats, and so it doesn't do that in humans, and so. In this case, it just has to do with how quickly the adducts that are formed are detoxified in the human. And so understanding, the rates of detoxification pathways can have a big impact on the outcome. So here is the example where, with those cotton rats, really, you sometimes have to use them because if you're going to be using a virus and you are relying on that host to allow it to replicate, well, if you use a species that doesn't allow it to replicate, then you're not going to get what you need. And that is why those hamsters and cotton rats are very permissive for those viruses, and they replicate very well in their cells. I don't know why, but they do. And so that's why you typically use those even that strain of rat, over, say, the Sprague-Dawley rat. So again, bottom line, you just got to pick the right animal model because again, with the thalidomide example, that's just one, and I'm sure there are many others, that can just lead to a lot of devastating outcomes, and you're wasting animals, and it just costs a lot of money, and you can really hurt people if you don't test it safely beforehand. So anyway, just pick the right animal model. Any questions?
I think it was good.
You're welcome.
Chris.
What's the difference between GLP and say CLIA or CAP, these different certifications?
What's CLIA? I'm not even familiar with what CLIA and CAP are, but I'll tell you what GLPs are, and then maybe you can tell me what the other ones are. So GLP, good laboratory practice, is a code of federal regulations that tells you the requirements that you need to meet in order for your study to be considered GLP compliant. And so it's really so that if anybody comes back to get your data, they can reproduce a study and know exactly what happened. What time was this animal dosed with what formulation, dosed by who, at what time. And so just so everything is accounted for and it's reproducible. Because it's basically a way to avoid, another situation like that industrial BioTest where basically data is falsified. So you can't. That's what the GLPs are. So what were the other ones? CLIA and CAP?
I'm not even sure what the acronym stands for. Clinical laboratory.
I don't know what the IA stands for either.
Yeah.
It's like when let's say you get your blood drawn in the hospital, your UAB, it gets sent to a lab to evaluate what your hemoglobin, your LDL, and all that kind of stuff. That lab is certified by CLIA.
Okay, yeah, so I imagine, whatever their requirements are that they need to meet, to be certified to draw blood and that the results are, again, I'm only speaking in hypothetical, I don't know,
It may depend on it's probably similar in nature, but GLP applies to
like the lab.
Non-clinical stuff.
Yeah.
Because as soon as you get into clinical stuff, I think, good clinical practice has to come into play.
Yeah,
and probably
consent
labs
and
have their own ways of, and I think it all comes down to assuring that things were operating and executed correctly, right? Yeah. So what's your favorite animal model?
My favorite animal model? Probably, well, I say favorite, but I'd say it's the easiest really would be the rat. But, I've had some studies with dogs, and so, they're sweet. They're the beagles, and they're super sweet. I think my least favorite is probably NHPs, just because I'm terrified.
Yeah.
So I usually observe everything kind of looking up through the window. I won't actually go in. But yeah, they all have their ups and downs. I think rabbits, if you're going to do a study where they can litter and have little kits, I've never done that, but that would be pretty cute.
Question.
You mentioned something in one of your slides concerning the toxicology results. At least you should have at least one toxic and one non-toxic. Okay. Is it always the case that the non-toxic level aligns with the therapeutic level? No.
If only.
No, not always. They have to do some calculations from. So whatever dose they give that has no adverse effect, and again, it's you may see something, but the question is whether it's adverse. So they go from that. It's called a NOAEL, like no observed adverse effect level, NOAEL, and that's the dose that you give, the lowest dose that causes no adverse effects. And so that's the dose you start with when selecting a dose for the clinic. You take that dose, swap it by 10. If you have a pediatric indication, swap that also by 10. So it just goes by factors of safety. So, it's not always perfect, but they try to keep it within a range to try to minimize any kind of potential toxicity in the clinic as best as possible.
I'm sure it's drug dependent, but do you have any idea what percentage of the time the animal models do not match what happens in humans?
That's a good question. I don't, but I know it happens. But I don't actually know the frequency. A lot of times, thankfully, they're mostly representative, but just really, I don't really have a target number to give you, but at least, I know that most of the time, at least one of the models that are used a representative. And there are cases where you see different things in your two models, and you're like, well, what do I do now? So then you have to kind of, and then that again really speaks to that weight of evidence approach. Taking all the data you have together and then trying to make an informed interpretation. But really, I can't think of too many times. But in those guidances I was mentioning, those ICH guidances, there are cases where they know that you shouldn't use an animal model for this. So, for example, a drug that acts through the HPA axis, rats are going to be more sensitive to that. Cats have a, if you're looking at something with the bladder or the renal system, don't use cats. I think just the way that they do it. I don't remember why, but don't use them. So, it's listed, this species is better for this. If you're going to use this kind of drug, don't use this species. So at least of the known ones. And I think as they come up over time, more knowledge is generated, the guidances are updated.
So I presume you guys also consult with a potential sponsor if they come in with a plan and it's not a good idea. I presume you get the feedback, or can they even come to you and say, we want to test this, why don't you guys tell us what to do?
Yeah. I mean, we do some of the upfront, help with study design for them, because there are cases where they come to us and they have no idea what they want. Mm-hmm. So we just help them give, ideas to try to, well, if you want to get this endpoint, why don't you just do, if you increase the number of animals, for example, perfect example, mm-hmm. Mice, as you know, are very small. Mm-hmm. They have about 0.5 ml of blood in their body. So we have sponsors that, well, we want to collect blood for this and that, and we're going to need 3 mils. I'm like, no, you're going to have to increase your number of animals. You're going to have to decrease the number of blood, decrease the amount of blood you need for your assay. But that's a common, the whole blood withdrawal thing, mm-hmm. and an amount of test article to give, or say by IV, you're limited by a certain volume, right? Mm-hmm. You don't give certain mils per kilogram by IV to an animal. So when they come and they're like, we want to give 30 mils per kilogram, I'm like, no, you can't. Yeah. So I mean, so you have to give some guidance, and I mean that's not that they they just aren't aware. But then there are some sponsors they're very on point. They're like, here's a study design, price it, do it. Okay. Yeah. So it really ranges.
Kelly was on a question.
Yeah, so in animal models, because space requirements, they're usually very sedentary.
So space requirements?
Yeah, because space requirements, animal models are very sedentary. So how do researchers account for that? Because usually when you're sedentary, you're more predisposed to certain disease conditions, and you're just testing them for things.
Yeah, I mean that's one of the. I mean, you can't. It's really, animals just aren't humans. You know what I mean? It's not something they account for. I think it's something that it is a part of those study designs. And I mean, for example, but not every species though. With dogs, you're required to exercise them. Otherwise, they're just that's, I think they get depressed or something like that. And that
just think about mouse models, because mouse models, mice are usually very active.
And, they're pretty active in those cages.
Feed them and just pumping stuff. There's food all the time. They're gonna have something. If you're gonna compare, mice in a cage versus mice in the wild. I mean, mice in the they may behave differently, but again, we're not out in the wild, so. I think, the level of activity isn't accounted for in terms of, that they're going to say, well, this mouse is active, this is inactive. And that's why you try to have a large enough sample size to get a consistent view as you go across the dose levels. It's not like a read across that you're trying to do, and at least because your control is that same, I'll say sedentary mouse that you're comparing to that high dose sedentary mouse, and the only thing that's different between them is the test article that you're giving. So, unless you see there are compounds that will cause lethargy, but then you need to be able to notice the difference with its concurrent control. So, but yeah, that isn't there. That and many other things you just can't account for just by the sheer nature of the fact that you're using animals to try to represent a human, which is why the strengths and weaknesses of characterizing your model is so important.
Any other questions? Maybe check chat box real quick. See if there's anything online.
Yeah, it's good.
Oh, sorry.
You do?
Hereby you have.
How much does the microbiota affect the toxicity screens from your animal models?
Unless we evaluate it. We've done studies where we've looked at microbiome. I don't think we actually got data back for that yet. But I mean, it may very well. I mean, there are conferences that are just focused solely on, you know, how that bacteria or how that internal system of microflora can have an impact, because they have drug metabolizing capabilities. So I mean, if you're, you know, there are studies where they would, I guess, get rid of, I'm not even sure what they gave, but essentially they made it products. Yeah. And then they would look at the difference in, you know, drug metabolism or disposition, and there were differences, but I mean, it's not always accounted for. It's just one of those things that kind of gets thrown in the bucket of, you know, yeah, even though the microbiome of a mouse or a rat is different from human, you know, I mean, if there were a case where it was, where you knew that it was going to have an impact, you know, there may be additional studies that, you know, like specific mechanistic studies or something you do. You know, every case is different.
And then in the field, do you see it where like the company will, whatever they're testing, they'll do it in their location and they'll do it somewhere else across the country just to make sure that, you know, it's following the same trend for reproducibility or anything like that?
Well, okay, so a couple of things to think about. One, these studies are very expensive. And so from the time that the company will patent their molecule, which is in that early drug discovery phase, that's when the clock starts ticking on the patent. And I think it's 20 years?
Yeah.
Thank goodness, yeah. 20 years, because after that 20 years period is up, other generic companies can come and make generics to compete with it. So, you know, while it may be nice to have, you know, lab over in Alabama do it and lab in California do, they look the same? Okay. Well, if they don't, first of all, it's, I don't know how often that happens because it just costs more money. But two, if the results are different, you're gonna have to explain that to the FDA. So it just gives you more to have to, not trying to say that you're trying to explain things away, but, you know, just kind of things to consider. Ideally, you know, you have everything done in one place. Doesn't just happen that way. Maybe not the way that Charles River is buying up everything, but, you know, I don't, if they elect to, they can, but I don't think it's very, very common.
I think it may differ too in what realm you're looking at for those animal studies. So in drug development and toxicology, when you use a reputable CRO like Southern Research, you would not, and Southern Research doesn't have a stake in the outcome of the study. You get paid no matter what, whether the drug fails or even it's toxic or not, doesn't matter for Southern Research. Where it does happen a lot is in early stage drug discovery. So for example, we have a project here in the Alabama Drug Discovery Alliance where we have really good animal data. We're talking to a pharmaceutical company right now that may want to license this. They want a little bit of the compound and run their own study in-house just to confirm the data, because we as an academic investigator have a vested interest in having a positive result of the study. Of course, we all assume everybody has integrity and, you know, there's not going to be any falsifying of data, but still, that's where that would really fall under the umbrella of due diligence. When somebody wants to pay a lot of money to license a molecule, you kind of probably want to do it 2 times, and maybe in your own lab will contract it out. So then it's done, but not with GLP tox is too expensive.
Yeah, hundreds of thousands of dollars for different studies. First study.
I think that's a irrelevant comment, but I think this is the patent law is a flaw. Because you should like, you know, I think if you do this, a lot of companies will rush to get a compound on the market and then ignore a lot of important studies.
Well, okay, so we have a lot of sponsors that'll come and they're like, you know, so it's called the IND, the investigation new drug submission, where you take all your preclinical data, all the data you have, and give it to the FDA. We get a lot of clients saying, I want to file my IND because it's September, and it's like June. So, you know, so I mean, they may rush us, but we still have to do the things that we're required to do. Otherwise, you know, it'll be written up as a patient, and you have to address it. So I mean, even if it pushes the time scale, it just makes things a little more stressful. But you still have to do everything that you're required to do, even if you're rushing.
All right, well, thank you so much. It was very informative. Thank you.